What is this we're learning in class?
I don't know, something about translation.
Well, I don't know anything about that.
Yeah, me neither.
Hey guys.
Hey.
Wait, guys, are you eating Chipotle?
Yeah, it's so good.
Are you trying to kill yourself? That stuff is so gross.
What do you mean?
Yeah, it's
Haven't
amazing.
you heard about it? There's like E. coli in it.
No, you're lying.
Yes, it's like all over the news.
Ew.
What would it do to us?
I mean, it could like undergo translation in your body and then release like toxic protein.
Okay, I'm done eating.
Nah.
I don't know how to do it.
Okay, Ashley.
Translation is a process by which the mRNA strand is read by a ribosome and encoded into proteins that help with the daily functions of living organisms. Both eukaryotes and prokaryotes undergo this process of translation. One example of a prokaryotic organism that undergoes translation is the E. coli bacterium. Translation of E. coli consists of 4 main steps. The first step is the charging of tRNA, where the tRNA is matched and attached to its respective amino acid. The next step is initiation, where the ribosome complex is formed. The third step is elongation, where mRNA is read through the ribosome and polypeptides are produced. The last step is termination, where translation ends and the amino acid chain is released.
The first step of initiation is the charging of tRNA with an amino acid FMET. To charge, tRNA synthetase is used to bind the amino acid to the tRNA. The Schindel-Gardner sequence, not present in eukaryotes, is upstream from the start codon AUG and helps to line up the small ribosome with the mRNA in E. coli. An initiation factor binds the small subunit to prevent the large subunit from binding. The charged tRNA binds to the start codon AUG on the mRNA at the P or peptidal site of the small ribosomal subunit. The large subunit binds to the mRNA to form the 70S initiation complex, which is represented by Pac-Man. All the while, GTP is the energy source that's promoting the process.
Now that initiation has occurred, elongation can happen. For elongation, a second tRNA binds to the A site of the ribosome, and then this causes the ribosome to translocate from the 5' to 3' direction. Then the E site, which now contains the original tRNA carrying the FMET amino acid, is kicked out into the cytoplasm, and the P site contains the second tRNA carrying the polypeptide chain. This process repeats itself until its stop codon is reached. It's important to keep in mind that the polypeptide chain is always located in the P site during elongation, and that the peptide bonds that are between each of the amino acids are formed by the 23S RNA acting as a ribozyme.
Now that elongation is over, termination is the last step. Once the ribosome reaches the stop codon, in this case UGA, it signals the end of translation. A release factor, represented by a ghost, binds to the stop codon at the A site, causing the release of the polypeptide chain. Translocation occurs again, in which the release factor is moved to the P site. The ribosome finally dissociates into its individual components.
3 hours later.
So,
No.
I'm
Struggling.
Dude, you're still here?
Yeah,
You've been here like all day.
it's my 3rd time.
How much Chipotle can you eat?
I like it.
You don't feel sick?
No,
Hey, good luck.
just kidding. Oh my god, I think I'm gonna be sick.
Now that we have a solid grasp on translation in E. coli, let's try our hand at a practice problem. Given this DNA template strand, transcribe it to find the mRNA sequence. Then, translate it to find the protein or amino acid sequence. Remember that cytosine pairs with guanine, guanine pairs with cytosine, adenine pairs with uracil instead of thymine, and thymine pairs with adenine for base pairing rules. Also remember that our mRNA sequence will be transcribed from 5' to 3' because that is the direction the RNA polymerases work. So let's start transcribing. The base pair of G is C, of C is G, of C again is G, of thiamine is adenine, of adenine is uracil, and of thiamine again it's adenine, and so on. Now that we have our mRNA sequence, let's start to translate it. To make our life easier, let's find the start codon AUG in our mRNA sequence. Here it is. That means everything before the start codon can just vanish because it won't be part of the protein sequence. Now that we have our start codon, let's divide the rest of this mRNA sequence into codons, which contain 3 base pairs each. So AUG is our first codon, CUG is our second, CGU is our third, AAG is our fourth, UAG is also our stop codon. Therefore, it's not going to code for a protein or an amino acid. So we can get rid of everything else to the right of the stop codon. And honestly, we can get rid of our stop codon since it's not going to be part of our amino acid sequence. So now that we have our amino acid sequence's mRNA strand, let's start building our sequence. So our first codon, AUG, is our start codon, so we can write FMET. Our second codon, CUG, let's look on this chart. CUG is leucine, abbreviated LEU. Our third codon, CGU, codes for C, GU, arginine, abbreviated ARG. And our last but not least, our fourth codon, AAG, codes for lysine, abbreviated LYS. So these are our amino acids, but remember, when we're building an amino acid sequence, we have to label our ends. So the side of the FMET is our NH2N, and the side of our final protein added to the sequence is our carboxylate end. This is our final protein sequence.